Good day everyone, my name is John Cedric Eshabal from PSY24. Today I'm going to make a clay model of the early development stages of an embryo. So these are the materials that I'm going to use. First is clay, which has different colors, and some toothpick. Without further ado, let's start. So first up is the two-cell stage of the embryonic development.
Cleavage in amphibians. Cleavage in most frog and salamander embryos is radially symmetrical and holoplastic, just like that of an echinoderm cleavage. The amphibian egg, however, contains much more yolk. This yolk, which is concentrated in the vegetal hemisphere, is an impediment to cleavage. Thus, the first division begins at the animal pole and slowly extends down into the vegetal region. It looks like I'm done with my two-cell stage cleavage of early embryonic development. So here is my comparison to the internet source that I found.
Next would be the 8-cell stage.
Cleavage can take place in two ways, either the holoblastic or total cleavage or the meroblastic, the partial cleavage. The type of cleavage depends on the amount of yolk in the eggs. In placental mammals such as humans, where nourishment is provided by the mother's body, the eggs have a very small amount of yolk and undergo the holoblastic cleavage. Other species, such as birds, with a lack of yolk in the egg to nourish the embryo during the development, undergo the meroblastic cleavage. Now that I'm done with the 8-cell stage, I'm ready now to move on to the next stage, which is the morula stage. Here is the comparison of my work and the internet source that I found. This model has a radial symmetry, a cleavage that is commonly found among echinoderms and Ophiuchus.
Next is the morula stage.
Morula, or mulberry in Latin, an early stage in post-fertilization development when cells have rapidly mitotically divided to produce a solid mass of cells that consists of 12 to 15 cells with a mulberry appearance. This stage is followed by formation of a cavity in the cellular mass blastocyst stage. A key event prior to morula's formation is compaction, where the 8-cell embryo undergo changes in cell morphology and cell-to-cell adhesion that initiates the formation of this solid ball of cells. Now that it's round and plump, let us compare it to the internet source. As you can see, the punctured holes represents the nucleus of the cells.
Let's now move on to the blastula.
Blastula is typically a spherical layer of cells, otherwise known as the blastoderm, which surrounds a fluid-filled cavity that is called the blastocele. Mammalian embryos form a new structure termed the blastocyst at this stage, which can be characterized by a mass within the cells that is obviously distinct from the outer blastula region. And now that we're done with the blastula model, let us have a discussion of the form of the blastula. As you can see on the internet source, we have the animal pole, the vegetal pole, and the blastocele. The cells in the animal pole divides quicker than that of the cells that is found among the vegetal pole region. And the reason why it is called the animal pole is because animals are livelier than that of the vegetative state of vegetables or plants, where the blastocele is the cavity-filled region of the blastula.
Lastly, we have the gastrula stage.
In gastrulation, the blastula proceeds to fold in upon itself to form those three distinct layers of embryonic cells. Each of these new layers is termed a germ layer, with each germ layer differentiating into a different organ system within the body. The three germ layers are called the mesoderm, the endoderm, and the ectoderm. The ectoderm initiates the formation of the nervous system and the layers of the skin. The mesoderm germ layer activates the formation of muscles and connective tissue inside the body. Finally, the endoderm layer starts the formation of the columnar cells that can be found inside the digestive system as well as many other internal organs. And now let's have an in-depth review of our model. We have the endoderm, the mesoderm, the ectoderm, the future anus, the blastocele, and lastly, the archenteron or the future gut. In endoderm or the internal layer, it creates the lung cells or the alveolar cell, the thyroid cells, the digestive cells, and the pancreatic cell. In mesoderm or the middle layer, it creates the cardiac muscle cells, skeletal muscle cells, tubule cells of the kidney, red blood cells, and the smooth cells in the gut. We also have the ectoderm or the external layer. Skin cells of the epidermis it creates, the neuron on brain, and the pigments on cells.